Good morning, students. This new part I am explaining to you now because till now we were talking about sex determination, sex-linked inheritance, Mendelian inheritance, multiple alleles, right? What happened is actually this is a continuation of Mendelian inheritance. Little bit, if you have an idea about chromosomes, I can explain you the next unit, that is linkage and crossing over. Now, what is this actually? As you already know that Mendel, when he crossed in a diabred cross, when he crossed two pure breeding plants for two different characters, he found that during the F1 gamete formation, the alleles undergo segregation. He did not explain which part is exactly responsible for the character transfer. He just said some unit factors are there, but chromosomal theory was given later after 30 years of his death by Sutton and Boveri. Right? According to them, whatever Mendel explained alleles, they are nothing but homologous chromosomes and the genes. Right? As he said, law of segregation, he said that they said that homologous chromosomes will separate during mitotic cell division. At the anaphase, they are pulled. Right? Then, next thing is about the law of independent assortment, where he said 4 types of gametes are formed, forming different combination: capital R, small s, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These are the gametes he told because of law of independent assortment. The same way actually the chromosomal arms behave, right? During meiosis, when they separate, they undergo crossing over, then they separate. They undergo separation and form independently assorted. Only thing Mendel explained was whatever happened was only in pupillans, and luckily for his luck, all the 7 characters what he explained, those characters possessing genes were located on 7 different chromosomes in pupillans. So this is what we call the luck factor, right? Therefore, the genes were not together, but later when he was present, he was not knowing anything about the chromosomes, DNA, nothing, right? Once the science advanced, biological investigations happened, then the word chromosome came into existence, what we discussed already. Chromosome having the number of alleles or the genes on it, right? Each chromosome will have large number of genes, right? According to his law, 7 characters, what he said, all those 7 characters were found on top 7 different chromosomes, therefore they were showing independent assortment. But in normal, all of us, in normal chromosomes, normally genes are located in a continuous sequential form. Suppose you think that this is a chromosome, diploid chromosome, this is one chromosome. Here you continuously have genes here, all these are called genes. Okay? If you think of gene A here, gene B here, they are so near to each other. Such genes will not undergo independent assortment during meiosis or during separation. During meiosis only, I think you would have heard, there will be gamete formation, right? Then if they are together found, then they will not undergo segregation or law of independent assortment. But the other way, imagine there is another DNA or another chromosome, one gene is here, one gene is, this may be the A, this may be ZX. This is found in another place, this is found in another place. Then they are far away from each other, right? That is the reason why, if the genes are located on the chromosome at a different length, different places, far away from each other, there are chances that they undergo crossing over. Right? Meiosis, you would have heard. In meiosis, we have learnt already, there will be pairing of the homologous chromosomes happening, and during meiosis phase 1, that is prophase 1, there is a big stage, there are a lot of stages. That is leptotin, zygotin, pachytin, diplotin, diagenesis. What happens in the first stage, leptotin, they will start separating. Zygotin, the paternal and maternal chromosome come together, right? That's a pairing of chromosomes, right? Later, they cross over and they exchange the genes. Then they separate off. So whenever they are crossing over, if this is the chromosome, then crossing over will not separate these 2 genes because they are very near to each other. But if this happens, another chromosome will be present over here, this will cross over like this. Okay? I hope you understood the difference. If this crosses over, together they go, right? This kind of arrangement of genes that are coupled together is called as linkage, right? Most of us, whenever I see a lot of students will have some kind of hitch for learning linkage and crossing over. The simple understanding is, don't worry about biology, think that normal English meaning. What is linkage? The two things that are joined are called linked words. Then crossing over and recombination. Crossing over results in recombination. Linkage and recombination you can call it. They cross over, new combinations are formed. Those are called recombinants. Even in Mendelian inheritance, you have learned 9:3:3:1 ratio. 9 and 1 are the parental combinations. 3 and 3 are the recombinant. They are the new combinations which were not there in the parents. Right? Now, here how and why these recombinants are formed, why sometimes this will not happen, that I want to deal with. Right? So, what you understood now by linkage? Linkage was the word first brought by scientist Morgan. T.H.Morgan. We call him the father of practical genetics, or he was the one or discovery genetics, new things he discovered. He worked on Drosophila. He is also called as father of fly because he worked on the organism Drosophila melanogaster. Right now, you don't worry about what I have written here. These things I will explain you. First, I want you to concentrate on linkage and crossing over, the name and why are the genes are linked and why are they called linked genes. Now, so what is that linkage then? If suppose this is the chromosome, okay, and genes are present one after the other, very much together, these are called as the linked genes. And the other way, if they are found one is here, then they are non-linked genes. Okay? They are not linked at all, they are far away from each other. Now, whenever they undergo crossing over, if these undergo crossing over, there is a chance that they go together, they will not separate. Therefore, if they go together, say maybe it is the color of the eye, this may be the height, they go together, color and height go together. So, parental characters will be retained in the offsprings more, right? But what about this combination? Here new characters are formed. Parental, both combinations are crossing over. Therefore, you are seeing some of the children will be exactly having some parental characters. Like one particular family, they have the fair color, their skin color is very fair and curly hair. That may go together in almost many of the children, you may get that. Right? Because that is called as a coupling, that's called the linked character, right? Same way, why they get linked is because they are found very much attached to or nearby each other, because one chromosome will have so big DNA and genes are units of the DNA. If these units are present one after the other, immediately attached to each other, they go together. But if they are present far away, then they don't relate, they get separated. Therefore, after Mendel, what T.H.Morgan, we call him also as father of modern genetics, because he was the one experimental genetics. We call him as father of experimental genetics, because he worked more in molecular biology. He has done a lot of work on this, right? So Morgan explained that linkage and crossing over. The genes which are linked will normally inherit together. Genes which are very far away from each other, they undergo crossing over, producing new recombinants. This is what his explanation. So what is linkage? Linkage is the ability of genes which are very much near to each other on the single chromosome inheriting together. That's called linkage. And if they are far away, then they undergo crossing over, there is no linkage, there will be recombinants. Right? What is the percentage of linkage? Depends on how far are they present. Linkage is how near. If they are very near, the probability of linkage is more. If they are little away from each other, the probability of them not to get linked is more. So more away from one another, the genes undergo more, they have the ability to undergo crossing over. This is how he explained linkage and crossing over. For explaining this, the linkage and crossing is over, he gave the example of Drosophila melanogaster with two different characters he considered. Right? That I am going to explain now for the linkage and crossing over. What did I tell you? If the characters are linked, then the parental combinations will be there in the children in more percentage. If the genes are far away from each other, then the parental combinations will be changed. Recombinants will be more. That's it. Right? So therefore I am giving you the example given over here. Just observe the examples carefully. Right? First you understand what is this meaning. Right? First cross A. What is this cross A showing? It is showing the parents with yellow white female. Yellow refers to the body of the Drosophila. White is its eye color. So two characters he has taken. One is the body color of the Drosophila, yellow color. White is its eye color. Actually, normally the regular type of Drosophila will have brown body and red eyes. This we call it as a wild type. Wild is the first one. Original one is wild. Right? So these are actually new varieties. We call them as mutants. Mutants are something newly originated. Right? So he considered the female, and actually these characters, one more thing he explained that some of these body characters are linked with the X chromosome only. They are called sex-linked characters. Means I am not talking about the autosomes. Those so many will be autosomes. No? Drosophila is easy because chromosome number 2 is equal to 8, right? So it is very easy for you to analyze this. Okay. Here what I said. This is a mutant variety. This is a wild variety. Two types of insects are crossed. Male is having brown body and red eyes. Means they have eye color only, red color. This is the male. We call this as a wild type. And female is yellow body with white eyes. This is a female. Okay. You just consider how and where are they present. See, this is the chromosome variety. First one X chromosome, second one is also X chromosome. And just observe carefully. I written small y, small w, small y, small w. These are the two genes that are present very near to each other. That's why I wanted to tell you. They are linked. Just wanted to show you. These are the linked genes present. They are present, see, so near to each other. Right? But I have written small y, small w because it is a mutant variety. It is a recessive character. So the white and yellow character is a recessive character. In females you have taken a female having yellow body and white eye color, which is a mutant variety and it's having recessive characters, and the genes Y and W are linked because they are side by side. Next one, brown and red eyed male you have taken. Brown eye and red eye, they are wild, means they are the original variety, they are dominant. So capital Y, capital W are written for the X, but Y chromosomes do not carry any genes because Y chromosomes are very small compared to X chromosomes. They are normally, in later again I will explain you, sex-linked inheritance separately. Right now you keep it in your mind that in males, X chromosomal genes are normally not present on the Y chromosome. Therefore, if the males are there, they will have only full genes on the X chromosome. Y is very small and it doesn't have any genes that are present on the X chromosome. Therefore, Y chromosome when I write, I don't write any alleles on that. Right? Y chromosomes do not carry any alleles that are present on the X chromosome. These are the so many things aspects you have to consider. First, I have told the Drosophila melanogaster. Its wild characters having brown eyes and the brown body and the red eyes. Mutant varieties are white eyes and the yellow body, right? And mutant varieties are recessive to the wild ones. And one more thing I told you, these are all characters of the body characters, the somatic characters, they are associated with the X chromosome only. Therefore, this will be called as an X-linked inheritance. And these genes are not present on the Y chromosome. So I have written the male with X and Y chromosome, but Y has no alleles on it. Okay? This is a parental cross. Now, when you cross them, the combinations are formed. What is the first combination? This, the first X chromosome combining with this, the brown-eyed X chromosome. X and X will fertilize. So this will be a female, but what is this? Capital Y, capital W is there, so it will become a female with brown and brown body and red eye. This is the wild type female form. Okay, it was not there. See, females were white and yellow, but now a new character is formed, that is brown body with a red eye. Wild female is formed. Next, what is this? Another X chromosome combines with Y. Small y, small w, X chromosome. Y has no alleles. This is a male having yellow and white, because yellow and white is recessive, but there is no another allele on the Y chromosome. This will be a male with yellow and white character. Okay. So these are the new F1 generation, the first generation. Okay. Now, don't worry about this, I'll tell you later. Till here only you observe. Next is in F2. F2, they undergo law of segregation. They combine independently. You would have seen, if you take capital R, capital Y, small y in the pea plants, capital R combines with capital Y, capital R combines with small y. Small y combines with capital Y, small r combines with capital Y, small r combines with small y also. Same way, all combinations happen and all they undergo different combinations. What are the combinations? First one, this one, small y, small w, that is the recessive gene for the yellow and white character, combining with the Y chromosome. This will become yellow-white male. Okay, this and this has combined. Next combination is small y, small w and small y, small w. This and this under one combination. This is again yellow and white. So yellow-white is the female parent, right? You can see here, female parent, what you have taken was yellow and white. But this is yellow-white is a recombinant, new character it was. Because males what you have taken is brown and red. But this is a new combination, right? Now, next, this one, that's capital Y, capital W with the Y chromosome. Capital Y, capital W, brown and red. This is the wild type. So this is again the parental combination, right? But what is this? This and capital Y, capital W, small y, small w, which is formed here. See, capital Y, capital W, small y, small w is a brown and red-eyed female. Now, observe which are all parental combinations here. What is the parent? Yellow-white female should be there. Where are the yellow-white female? There is one yellow-white female here. Then, what is the other parent? Brown-red is male. 1 and 2 are the parents. These two are recombinants. Then when you observe the ratio of parental combinations and the new recombinants, it comes to this. That is 98.7% are parental combinations. 98.7% a parent means what? These parents, yellow and white females and brown and red males, they are 98.7, means parental combinations, their genes or alleles are not separated, they are together. That is 98.7%. But recombinants also I told you, there is one recombinant, there is another recombinant. The recombinants are only 1.3%. This indicates that the genes were together, they were linked, therefore recombinants are produced less. This whatever I drawn is called as the chromosomal mapping, gene mapping. Genes, where are they located? I have given the positions. This is called the gene mapping. This is a very important thing. Further again, I told you earlier, molecular biology when I explain, again you will come to understand, know where are they, what are the genes, what are the alleles and all. But first you get to know all this. So whatever I have given here as the parental combinations and the recombinants. This is the ratio of F2. The F2 ratio I have given you. This is the combination. So this complete combination you take it down. Carefully you write down, because some of the books also are given so confusing. They are written Y plus, W plus, don't write. Right? Capital W, capital Y is enough to write the wild parents. Okay? Write it down properly now. Right? This is what I am expecting now you to do. Write it carefully. You take 2 more minutes and write it properly. I hope you are done it. Okay. Now let us observe the second combination. Right? What is the second cross? He has taken another character in Drosophila only. It is the white eye color with a miniature wing size. Wing size, these insects, I told you Drosophila normally are produced during the fruiting season. Whenever the fruits are formed, when they start rotting, the fruit flies will come. No, those are nothing but Drosophila. Therefore, they have certain wings. If this is their body, okay, their body, there will be wings. This, their wings. Okay, this is the broad wings. Another one is the one having miniature wings. Two characters taken, right? This is the miniature wing, and this is the broad wing, right? First one I have written you parental combination white with a miniature wing in females. Means here it is white eye color, miniature wing. See, you can see the differences. They are far away from each other. The genes are located. This is the gene mapping. So female is homozygous with XX chromosomes having small y, small w, small m, showing that they are white-eyed with miniature wings, small wings. This is small wing I showed you. It was crossed with red-eyed, broad-winged character. Broad means widened, right? So the red-eyed and broad-winged character is a dominating character, and it is called a wild character. Whenever I say wild, they are the first formed characters. See, I am making you understand this clearly. Later again, you see the NCERT book, you will understand how to write it. Some of the things are confusing, means you just clarify now only. So here the genes are written, 1W for white color is here. This is M for the miniature wing. But what about red and broad? Red-eyed character is capital W and broad wing is M. So they are also far away in X, and Y chromosome has no genes, no alleles which are present on the X chromosome. Now in F1 generation, I am writing this in the F1 generation. So you have to write all combinations. The small w small m combining with the capital W capital M. This is the recombinant. F1 hybrids are recombined ones, right? This is a red with a broad wing female. Actually female parent what you have taken is only white eye with a miniature wings. But now we have got red and the broad, which is the wild character in females. Now another male what you have got is this small w small m with the Y chromosome. This is again white and miniature male. White and miniature males are not there. This is also new recombinant. Now if you take F2, this is second generation. What I am writing here is this is F1, F2 is here. Whenever you write F2, you can see that all combinations are formed. See, first one w small m for the white and miniature wing combined with small w small m, which is the white miniature female, which is the parental combination. See, parent also is white and miniature wing only. This is the parental combination formed here, right? Next is capital W. See here, this capital W capital M with a small w small m formed another combination. This is red and brown. It's a female, right? It is a recombinant. Again, one more parental combination formed, capital W capital M with the Y chromosome. This was a red, that's a wild type. Red with the broad wings as in the wild varieties, and this is parental combination. Another combination, this one small w small m along with the Y chromosome, which is white and miniature male. Then who are all parents now? Parents which are checked. White miniature was the female. White miniature females, how many? Where are they? White miniature female, this is the parental combination. Another parent is red and broad, which is male. Red and broad male, where it is present, this is the parent here, red and broad male. So these are the parental combinations. In this combination, parental combination gets 62.8% are the parental combinations, right? But recombinants, what you are getting here, it is around 37 point, this is 62 point, okay, 37.2. This is the recombinant, okay. Some percentage will come at the end. So it is 37.2% 2% which is recombinant, and this is the parental combination. You can see how much is different. See, this parental combination has 98.7%. Here parental combination is reduced, new recombinants are formed, this number is increasing. This means genes are far away. They are non-linked genes, they are not linked at all. They are very far away from each other. So crossing over has happened more. Therefore, we are getting new recombinants in 37.2%. Some more end will be there. Means 2, 3 or something like that. This is 62.87 and all is there. But anyway, percentage you observe. This percentage will give this. You have to try out also. What is the first combination percentage? Parental combination is 98.7 parent combination, but recombination are hardly 1.3%. But in this you observe parental combination is 62.8 and recombinant percentage is 37.2. Means more recombinants, indicating more crossing over has happened because genes are away from each other. If they were together, then you would have got parents more in number. Parental possibilities were more. Here, as they are far away, recombinants are more in number. So understand that this is a beautiful subject. Observe, read, write, understand, ask me questions. This is what is the opportunity for you to understand. Again, if you are not understood, again it starts, I will explain you again. Right? But I want you to get into these crosses. You should write this crosses. It's a 5-mark question. Write the cross and explain linkage and crossing over, linkage and recombinants in the Drosophila melanogaster given by Thomas Hunt Morgan. T.H.Morgan is the name of scientist, Thomas Hunt Morgan. Okay, this you have to be perfect because this is the part where most of the students will leave their questions. But I am telling you, this is the point where everybody say it's difficult, you study that first. Then and there is nothing called difficult in the whole of syllabus. Anywhere nothing will be difficult unless you are understanding it. So just understand, it becomes simple to you, right? Fine. So I want you to take this down also. Take it down, the combinations. Okay? First cross and second cross. Cross A, cross B, both. So once you are done with this, let's now move on to the next part, that is sex-linked inheritance. Linkage, I gave you some background, right? So now I will give you the real details of the sex linkage. What do you mean by linked inheritance and how the sex linkage happens and what is the feature if it is linked, what is going to happen. Okay? So let me give you the details of sex-linked inheritance. Okay? So as I told you, most of the things in genetics, in modern genetics, were conducted by the father of experimental genetics. Who is that? Scientist T.H.Morgan, right? So I am talking about T.H.Morgan's next discovery. You will understand now how exactly, with one human example I give, then you are able to follow it easily, right? So what is that? I will give you the now next unit, that is named as sex-linked inheritance. Okay? So what do you mean by this? Till now, I just gave you most of the genes from the body characteristics. If they are linked with the sex chromosome, they are called sex-linked genes, and that inheritance is called sex-linked inheritance. Okay. Now, what type of characters are linked with the sex chromosome? So imagine there are two chromosomes. You already know. Human females will have one X chromosome and another X chromosome. This is sex chromosome. This is the female character. But when you think of males, they will have one X chromosome and one very small Y chromosome. This is Y chromosome. So why I am saying all this? Because see, both the X chromosomes are homologous. They have the same genes in all of them. But in thinking of males, these are the males, they have X chromosome, okay, big. Only till here few genes are there. Suppose any genes are located here, that part either homozygosity or heterozygosity is not observed in Y chromosome. Y is a very small chromosome. Now, if any character for the body, if it is linked with the X chromosome, imagine there is one character gene present for the human, some character. Let me talk about the disease, which if at all the genes are located on X chromosome for any other body characters, we don't mind. But there are certain characters or the genes that are linked with the X chromosome, but they cause certain disorders, genetic diseases. One of the example is hemophilia. I'll explain with the same example. Hemophilia, I'm explaining you. Okay, with this gene. Before that, let me give you the details of hemophilia. So, what is hemophilia? It is a kind of genetic disorder in which the person who is affected or the patient will not clot the blood in any small injury also. Means there is a lack of blood clotting factor. So this disease is characterized by the absence of or lack of blood clotting gene. But blood clotting gene is absent. Okay. This is the reason why any smaller wound, small pinch also will result in continuous bleeding for more than half an hour or 1 hour. Very small wounds. But normally in our blood, if at all we get a wound, within 3 to 5 minutes it will block or it will clot and bleeding will stop, depending upon the size of the wound. If the wound is very small, it will heal up very fast. Suppose one pin, you get a pin, just poked, within no time, 1 second, just become over, right? But with hemophilic patients, it will not happen. Hemophilia is the kind of disorder, genetic disorder, in which the patient suffers from excessive bleeding from any wound. Sometimes they bleed to death. This is very important for you. They will die because of over bleeding, hemorrhage happens, right? If you have seen any hemophilic patients, then you will understand what it is. Now, next part I want to explain is where was this hemophilia first identified. Hemophilia disorder was identified first in the royal families of Queen Victoria. See, so many words you have to remember. So this is the first point, the second point. It was identified in Queen Victoria family, royal families of England. She was having the gene for hemophilia, right? And she was not showing the character or she was not showing the bleeding problem. But what happened was, she had 2 sons who died in separate accidents or some problem happened. They died because of excessive bleeding. Means smaller wound or bigger wound, they died because of excessive bleeding. That is the time when scientists, all medical people started studying why this would have happened. Then they came to know that those boys, sons of Queen Victoria, lacked the blood clotting factor. Then they traced it. They came to know that this character of non-clotting or non-clotting character was present in Queen with the female, the woman, the mother, which has transmitted to the children. Therefore, they declared it as a genetic disease. And this, because of this character or this discovery in the royal families of England, it is called the royal disease. Hemophilia is called as a royal disease, and it is also called as bleeder's disease, because they will bleed. Bleeder's disease. Okay, remember. Now, keeping this in mind, I will go to the details of this. So, hemophilia is a disease of genetic type which results in the excessive bleeding from any wounds due to the lack of blood clotting factor. It was discovered in this family, but nowadays it is found in every other family also. Because it was first discovered here, it is called the royal disease. Okay. Now, coming to this, imagine the diseased character. It's a hemophilia. This will be always disease. This disease is always recessive in nature. Most of the genetic diseases are not dominant. They are due to the recessive alleles. Therefore, if suppose the males or the females have normal allele, or if they have these alleles, no? If they are homozygous, then if they contain the capital H gene, which is for the normal blood clotting, and if they have a small h for hemophilic character. If this is a female having heterozygosity for the normal allele and another gene will be, or another allele will be for hemophilic gene. She is heterozygous for the normal character. She will appear healthy. She is the healthy female. Why? Because always I told you, diseased genes are recessive to normal genes. So capital H on X is a normal gene or healthy gene. X with small h is a hemophilic gene. Okay? Such females will not show any hemophilic character. They look normal. But suppose the men, they contain the diseased gene small h. What happens if these contain small h? There is no another allele for them to overcome the disease. Because Y is so small, they will be suffering. Such males are normally hemophilic males. Why I told you probability is more in males? Because they will have only X chromosome. If they get healthy capital H here, they are not suffering. But probability is more for them to get such diseases like hemophilia. Females, see, on the other hand, females will get capital H with small h. They don't show any symptom, they don't have the problem at all. They look normal. Such females who contain the hemophilic gene in a heterozygous condition are called carriers of that disease. What is the meaning of carrier? They carry the hemophilic gene or the genetic diseased gene from them to the next generation. They transmit to their sons. Therefore, they are normally called as the carriers. Again, I will tell you when I explain you the disease details of inheritance. Now, with this, what you can understand is what is hemophilia. It is a disease, genetically transmitted disease. It's not a sexually transmitting. Remember, don't mix them. It is a genetically transmitted disease. It is caused due to the problematic blood clotting gene which is absent in the hemophilic patients. It was discovered in royal family of England. Then apart from that, this disease is transmitting, or probabilities are more in males to express than females. Okay. Now, you may ask me, small h comes, that also will be a female. Females will get disease. Sure, they will get disease. But remember, if the female get hemophilia, she get hemophilic, she get that disease. Then the baby which is born will be normal. But when she get the onset of menstruation, menarche when she gets it, she will bleed to death. So, such females cannot survive practically till the age of reproduction. That is the reason why females with hemophilia are not there in literal terms. They are not going to survive. Males can survive because they may have some bleeding very rarely. Normally, bleeding happens only when their milk teeth get replaced with the permanent teeth. That's the time when they will get, sometimes the bleeding, continuously bleeding will be there, and after that they will not have to lose any blood, because that is not a major characteristic of men. Men will not normally have the bleeding in their life unless and until they only meet with some accidents. Otherwise, but females are, it is like a compulsion for them. For every menstruation they lose blood, right? Therefore, they may not survive with disease. Men can survive with hemophilia. Okay? Keeping this in your mind as a background, let me give you what kind of parents inherit hemophilia, how they inherit, what is the pattern of inheritance for sex-linked genes. This is called sex-linked inheritance or X-linked also. This is not only sex-linked, it is actually called as the X-linked linked inheritance. Okay. Now let us continue with the inheritance of hemophilia. How it is inherited from parents to offsprings. Okay. Let us talk about, I told you the example 1 and video, it is hemophilia, right? This is example 1, hemophilia. Okay. Let us consider the female who are totally normal, non-hemophilic, right? So, the parental genes I am taking, parents. What is the phenotype I am considering? Phenotype we are considering are the healthy females, non-hemophilic and healthy females. If she is married to the hemophilic man, hemophilic male, okay, then let me write about the genotypes. Now it is not a simple genotype, remember, you have to write the complete chromosome and the genes, okay, fine. So what you will write here, healthy females will have X capital H, X capital H. See, this is the genotype. Don't actually circle these, only I am just showing you because you will not confuse, you should not confuse. X capital H referring X chromosome having the allele capital H, X chromosome having the allele capital H. Now, the hemophilic males. So, I will write the hemophilic gene with the red color. Otherwise, you will not distinguish. They will have X with small h and Y chromosome. Y don't carry any genes. So, Y I will write normal with a normal color only. Okay. This is a cross. Don't mix with this X. Okay. This is the genotype of the healthy female without hemophilia, because I told you hemophilic allele always is dominant over capital H is dominant over small h, or X capital H is dominant over small h. Right? But she will be a normal female. He is a hemophilic male because he contains the gene for hemophilia. Now, while writing gametic generation, females as already they have homozygosity, they produce single type of gametes, X with capital H, circle line, right? Then crossed with the gametes of males I am writing now. X small h will segregate from Y. Okay, this is the Y chromosome. Okay. Now the sperms, these are the sperms, I hope you know, these are sperms. If this sperm fertilizes the egg, then it will be X capital H, and how is another one? X small h. Right? So just to distinguish I am writing this is with the red color. So such females, you can observe here, she is having one capital H gene allele, one small h. But I told you capital H is dominant over small h. Capital H is for normal blood clotting. So she will be healthy, normal female. Healthy female. But genetically she is carrying the, genotypically she is not totally normal. She has the gene allele for hemophilia. Therefore, such females are called as carrier females. You are writing this in the bracket, right? Because carrier females you have to write in the bracket because they don't appear as if they have hemophilia. They don't have the blood clotting problem. They are normal. Therefore, they look healthy, but they are carriers for the diseased gene. Now, if this is a fertilization, second fertilization, X capital H and Y. This is the male, right? Y chromosome has come, this is a male, but he has capital H from the mother. Although father had the hemophilic gene, he did not get the X chromosome from the father, he got the X chromosome from the mother. Therefore, he looks normal, he is healthy male. So, he got the healthy gene from his mother. Therefore, both in F1 or children produced from the parents with healthy or non-hemophilic female and hemophilic male, the children are both normal. They are not having any disease, right? But females are carrying the allele. Okay. Now, what happens such females who are carriers if they are married to a normal healthy man? Okay. I am writing here F1 what females or F1 females marrying to normal, healthy male. Okay? What is the phenotype of F1 females? Now you tell me. Phenotype of F1 healthy females, phenotypically they are totally, I'm sorry, they are healthy females, but they are carriers. I am writing in the bracket carrier. Remember. They are marrying to healthy man. Healthy man means they are totally normal, healthy man. So don't write the cross here. Always I see students doing that. These are the children born from the same parents, right? They are the siblings, brothers and sisters, right? I am saying that is the reason whenever you write human example, don't have to give direct crosses and all, don't write. Write that if they are married, okay? And this kind of female from F1 hybrid, if she marries another healthy man, I am saying, right? Don't write cross areas, healthy man, healthy woman, no. These are the siblings, brothers and sisters there, okay? Now, therefore, whenever you write human example, don't write sibling crosses, right? Now, phenotype is healthy female and she is marrying another healthy male. What is their genotype? This female has X capital H1. See, she has one X capital H. What else she has is another X small h. This is very important. This she will not show. She is a carrier. She won't show that she is having disease. But she will be having diseased gene with her. Now, marrying to healthy man X capital H and Y. Okay. Now, what is the gametic generation? The female now become heterogametic. She will produce two types of gametes. X capital H one gamete, then X small h another gamete. Now what about the males? They anyway heterogametic. They are X capital H and Y chromosome. Now you put the checkerboard and analyze this. Right? I'll just write the checkerboard for this. You have to remember while writing checkerboard, males and female is very important in this cross. Right? This kind of F2 generation. Bullet checkerboard you are writing. One side I am writing males, one side I am writing females. Very important is you have to write males only. Male means you have to write this X capital H gamete, X capital H, and here is the Y chromosome. Here is X capital H and known as X small h. See, wherever I am writing, I am circling the gametes. Combine them. What is the combination? X capital H, X capital H. How is this female? She is a normal, healthy female. Now, X capital H and Y, he is the healthy male. What is next? This is X capital H. X capital H and another X small h. What about this? She is also a healthy female, but she is a carrier genotypically. Last one is important. You have X small h and Y chromosome. He will be affected. This is the hemophilic male. Hemophilic male. Okay. So now analyze the result in F2. What happened in F2? Among the four possibilities, the females are right. How is the female generation here? Okay, let me write the females of F2. How are they? One female, second female, both are normal. So 100% healthy females. Okay, that's in the F2 ratio. What about males? Males, one is healthy, one is hemophilic. Therefore, male ratio is 1:1. That is, one will be normal, one will be hemophilic. Did you understand this? This crossing you have to write with X and H only. You can just write X and Y, no. Right? You can't write only H and small h. You have to write along with the genes only. Some books are given X dash and all, don't do that. Write X capital H small h only, because dash sometimes you will forget also. Therefore, better you take up the genes and alleles properly and write the cross. So, what are you have to write is, start with the first one, inheritance of hemophilia. What happens? You have taken healthy female and the hemophilic male. Then healthy female means she is non-hemophilic. She will have X capital H crossed with the hemophilic man having the hemophilic gene X small h and Y. He is hemophilic. He has hemophilia. Then gametic generation, she is homozygous and homogametic. She will produce only one type of X, it will be X capital H. But men are heterogametic. He will produce two gametes. One is having X small h, one is Y chromosome. Next, during crossing, the sperm may fertilize the egg with a normal type, and this will be a healthy female with X capital H, X small h, because capital H, X capital H or the hemophilic character is dominant over the hemophilic is recessive to the healthy character. See, this is hemophilic, this is the healthy gene. Now, she will be female, but she is a carrier, but she looks normal. So, healthy female. But what about the next combination? X capital H Y will be producing healthy males, because this boy will get the X chromosome from his mother, not from the father. He is getting from father only Y chromosome. So he is getting maleness from father, but X chromosome is getting from the mother. He is a healthy male. Means, although parents, among the parents, father is having diseased gene, in first generation, no child is affected with hemophilia. Remember this. Now, if such female who is healthy but carrier female, she gets married to a healthy man, right? Phenotype, I am writing healthy woman with the carrier character with a healthy man. Genotype is X capital H, X small h, because she is carrying the gene. She is a carrier crossed with a normal character. Then she produced two gametes because now she is carrying one new allele, which is hemophilic allele. She undergoes segregation. He will anyway produce two gametes. Then one side I am writing male gametes X capital H and Y. One side I am writing female gametes X capital H and X small h. Fertilization, this and this combination X capital H producing healthy female, X capital H and Y producing healthy male, X capital H small h is producing healthy female but they are carriers, but X small h and Y is producing hemophilic male. Now, I told you the ratio. How is the ratio? The females are, see, this is also female, all are 100% healthy. But what about males? 50% males are hemophilic, 50% are normal. Therefore, you are getting 1:1 ratio. Got it? You take the complete chart now. Then I will analyze the details of this cross. Okay? Fine. Take it down. Have you taken down this complete table? I had started with the P1 generation, F1 and F2 with the checkerboard, and at the end I give you the various probabilities of children having hemophilia. Now you understand one kind of pattern of inheritance here, how it is inheriting. So here the disease was there in first generation. He is hemophilic man, right? It is there in him. But in F1, no, nobody got it. In first generation, so parents it is there. In F1, nobody has got it. In F2, again you have got a hemophilia. F2, again it is expressive. Right? So, how it is inheriting? Right? If it is present in P1, let me write here, again overall analysis. If it is found in P1, if it is not there in F1, it is present, it is not there in F1. It reappears in F2. But in F3 it will disappear. This is the reason why we call all this sex-linked diseases or sex-linked inheritance as skip generation inheritance. Generation inheritance. That is, it is skipping a generation every time. It is not continuous. It skips a generation and next generation appears. First generation is there, next generation it is not showing any symptoms. Third generation it again comes. Fourth generation it disappears. This is called the skip generation inheritance. But observe how is the pattern of inheritance. right? Let me write here. How is the pattern? Imagine it's P1 with male and female, F1 male and female, F2 male and female. I am writing all of them. F3 male and female, F4 male and female. Suppose this male has the diseased gene here. Okay, this gene will move to the, see, the father had it, he has transferred it to his daughter. The gene will go to the daughter. Okay. Now, from this daughter, when she gets married, she will pass it to one of the son. This is again going to the, from mother to the son. That is female to the male, gene is here. Again male to the female, again female to the male. This is showing, you can see zigzag pattern, right? Male of one generation to female of next generation, female of that generation to the male of the next generation, and so on, male to female in successive generations. Therefore, we call this as crisscross inheritance. Remember, crisscross we call it inheritance, right? That is zigzag inheritance. That is, it is inheriting from father to the daughter, daughter to the son, son to the daughter, like that continuously. What you have to learn by this? If the disease were there in parent, it comes to the daughters, but daughters are not showing any symptoms of this. This is the reason why in most of the marriages, the female will remain looking healthy, but internally her genes are affected. Those genes are transmitted to the next generation. Therefore, it is reappearing in the third generation. Therefore, females will remain like a silent carriers of the disease. Right? Understand this very much, because skip generation inheritance meaning is different, crisscross is different. So, sex-linked inheritance itself is called as either skip generation inheritance or crisscross inheritance. Then, what is their meaning? The sex-linked inheritance, whenever it is transmitting the genetically transferred diseases of the characters, if it appears in one generation, it skips the next generation. It won't appear in the next generation. Again reappears in the third generation. Again skips the fourth generation. Therefore, we call this as skip generation inheritance. But what is the next thing? Crisscross inheritance. What is crisscross? If in parental generation it is found in males, in F1 generation it comes to the female. The gene will transfer from males of one generation to the female of next generation. Then from that female it will go to the third generation's male. Then male to the female of fourth generation. Like that it transfers in a zigzag fashion or crisscross fashion. Therefore we call it as a crisscross type of inheritance. Remember this very much, because crisscross inheritance can be asked for 1 mark, skip generation inheritance can be asked for 1 mark. What is hemophilia? A lot of questions will appear in this. Who are the female carriers? Why males have the probability of getting this disease? Very important. Because males are having X chromosome and a very small Y chromosome. X chromosome, if it is healthy, they are healthy. X chromosome is diseased, they are diseased. Therefore, the probability of males getting sex-linked inheritance or sex-linked diseases is more than females getting the disease. Did you understand? Analyze it. So, after this, we have to start with the next example, that is color blindness. First, you understand this. Write 1 or 2 sentences, that will be your homework. Some of the questions I will give, you write them down. What is the first one? What do you mean by sex-linked inheritance? Okay, written. Second, what is the meaning of skip generation inheritance? Third one, what is crisscross inheritance? Fourth one, who are called as carriers in sex-linked inheritance? Okay, these major things. Then from linkage, what do you mean by linkage? What is recombination? What is crossing over? Right? These things you understand thoroughly. 1 mark. Let me see how many of you write and show me. Okay? Now we'll move on to the second example. Okay, let's now move on to the second example. as already I gave you first example as hemophilia. Second example in humans that follow sex-linked inheritance or that will inherit through sex chromosomes is colorblindness. What is the meaning of colorblindness? It is simple meaning. Some people are blind for certain colors. They are not blinds. Certain colors they are unable to distinguish. That's a meaning. That doesn't mean that all blinds are this. No. They can see everything normally, but certain colors they cannot distinguish. Right? For example, some cannot see red color as red color only. They will. It looks like some orangish color. Right? Some will not see greenish as green color, somewhat bluish color, some other color it looks like. So therefore they are the people who are called color blind, right? So color blindness is of different types, right? Somehow red blindness, we call it as protanopia. Protanopia is the red blindness. Some will have green blindness. Green blindness is called deuteranopia. Okay, but very important thing is some will not be able to distinguish between red and green color. It is called as the red-green blindness, or its name is daltonism. Red and green differentiation you cannot make it. It is called as daltonism, right? You are not able to identify the difference between red and green color. They look alike. No. The question is, if you have this red and green blindness, it is very crucial in your life, because whenever you go to get the driving license, there will be one question: do you have color blindness? That will be a question. Or do you have red-green blindness? That will be a question. Most of the students will write. Whoever go to get the license, they say no. That is like mandatory. But hardly anybody have the idea that it has to be checked also, right? Actually, there will be earlier they used to issue license to you, driving license. Then they used to have a chart, and in that chart they used to have red, green and orange colored dots, right? Like there used to be a chart like this, big chart, imagine, and there were some dots in this, right? Some of the red dots used to be there, red dots, okay? You have to identify how many red dots are there in this and how many green dots are there in this, right? Can you really identify them or not? This was the earlier way to identify whether you have red or green blindness or you don't have, or whatever it is. This is how they used to test earlier. Means identify how many red balls are there, how many green balls are there. If you have red, now also you can see, in this only you can identify how many red balls and how many green balls. And if you have identified properly, that's why I written red and green only, then you must be normal if you are able to identify. And so many times I mix with some other colors also, like blue I am mixing. If you are able to mix this also with the green, then something is wrong with your identifying colors, right? You have to count how many red. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 red are there. Remaining green you have to count and write it. That shows you don't have red-green color blindness. Very important, and this has to be issued that this person is having no red-green blindness, and that should be certified. Then only you are eligible to get the license. Why? Driving license you get only if you are not color blind. Why? Because you know it, right? What is that? If you go to drive vehicle, if you are in the signal, the signal colors are red and green. If you are not able to distinguish between red and green color, you are gone, right? Therefore, it is very much important for you to follow certain rules which are already made. Rules are not made for the joke. Most of the time, now also people will joke. Why the lockdown is extended? It's not because somebody wants it. You people want your safety, you stay home. It's not a rule. It is your safety. You are given just open environment for you to stay away from the disease. That's not a rule. It is your safety. Why to wear mask? Your safety. Why to wear helmet? Your safety. If police is not there, you will not wear helmet to show the hair or anything like that. These are all. What happens is, most of us, we think that whatever is made as a rule, you have to break it. Wrong. You will have to make rules to understand that they are for you. Your safety. Follow the rules. Okay. So this is the red-green blindness called daltonism. Now I'll explain you its inheritance. Okay. Right. How it is inherited? So I hope you are counting this. You are counting, right? Count and write it. Now let me give you its inheritance. How it is inherited? Almost similar to what it is here. Hemophilia. Hemophilia, how it was inherited, same way it is inherited also. Right? Now let me write the genes as X capital C as normal visioned person. Normal vision means he don't have blindness. X small c as the color blind person. He is a color blind man or woman, whatever it is. Color blind gene. This is one allele for normal vision. This is allele for color blindness. Right. T1 generation I will write. How we are selecting? You can take the parents vice versa also. Maybe female only is color blind. You can take that way also. Then the things will change. So let me take parental generation. The color blind female. Color blind woman. See, don't take color blind hemophilic female. Right? Sorry, hemophilic female is not there. Color blind female can exist. Right? Then marrying with the normal visioned man. Right? This is a phenotype. What is the genotype of this? Genotype is X capital C. She is color blind. She should be homozygous X small c, X small c. Okay? You can write anyway. Anyway you can take the cross. Okay? And normal male X capital C and Y. In Y you are not supposed to write any allele because Y chromosome do not contain any alleles which are similar to X chromosome. Now during gametic generation, gamete formation, the females produce single type of gametes in this cross. Right? Males will produce two types of gametes, X capital C 50%, Y 50%. Now the sperm with X capital C fertilizes the egg. It becomes X capital C, X small c. This is a female who is normal visioned, but she is a carrier. Where she got the gene now? She got the allele for color blindness from her mother only. Now how about the male X small c and Y? This will be the color blind male. Why? Because he got the color blind gene from his mother. Right? This is the major problem happening. So although his father is normal and mother has color blindness. Now, if this female who looks normal get married to the normal visioned male, what happens? So F1 females getting married to the normal visioned male, what happens? So how is the phenotype of F1 female? She is normal visioned female married to normal visioned male. Okay, two things. What is the genotype now? Whenever you write genotype, normal visioned female should be XC then. X capital C, X small c. Right? Cross with normal visioned male, X capital C, Y. Now, during gamete formation, she will produce two gametes, X capital C, X small c. He will produce two gametes, X capital C, Y. Okay. Let me put the checkerboard and analyze F2 generation. X, right, first now I will write the checkerboard. You write a bigger checkerboard. I don't have place much, so I am writing smaller one. Okay. This side males, X capital C and Y I am writing. This side females I am writing. This side it is X capital C and X small c. Then cross them. X capital C, X capital C. How is this? This female is normal vision female. Then X capital C, Y. Always C, you show a small tail like this. Otherwise, you will mix up both the C alphabets. This is again normal visioned male. Then X capital C, X small c. This is a normal visioned female, but she is a carrier. Then X small c, Y. He is a colorblind boy. Colorblind male. Okay. So in F2 generation, what you got? The females are 100% normal vision, and males have got 1:1 colorblind, 1 normal. Right? Again, you can see zigzag type of inheritance. In the first generation, you got the disease. Second generation, it has transferred from this female to the male. Right? Colorblind. She is a female colorblind. And if I am talking about if they both are normal, how it is? Colorblind male, female getting married to normal male, you get colorblind male because female is transmitting. Then if she looks normal but she is a carrier, then she will transmit to one of her son. Again, zigzag type of inheritance. One generation it appears, one generation disappears. But here you have taken diseased one only, diseased female, therefore it is transmitting like this directly. Right? You can take the same way how I took for the hemophilia also. What I took hemophilia, we are taking the female who is normal, male is colorblind. Then F1 generation, no one is affected. F2 generation, among all the generations formed, all the offspring produced, 75% are normal, 25% are diseased. Among them, males are more diseased, having more chances to get affected than the females. So that is the color blindness. You can take another type of cross also in this. Okay, color blindness. Understood? Right? You solve these and explain me. Now I told you this. I told you the cross. You write another cross. That's a normal female with a colorblind male. Colorblind male. You take it and write the cross till F2 and show me. Okay. Now the last example I give you. That is about the third example. It is about the eye color in Drosophila. So remember when I told you something about Morgan, T.H.Morgan, Thomas Morgan during linkage and crossing over, I mentioned that some mutants are produced in Drosophila melanogaster. Now I am giving you that example. Example number 3, that is the eye color in Drosophila melanogaster. Okay. I told you this is the fruit fly, right? Fruit fly, and its chromosome number 2n is equal to 8. So very simple to work on with this, right? And some percentage of chromosomal genes of Drosophila and humans are matching also. Therefore, this is the most important insect, and most of the geneticists who want to work on this. This is called as the Cinderella of genetics. Cinderella, they call it Cinderella of genetics because it's like pet for everybody to work on, right? Anyway, why I talked about Drosophila? This was actually the whole sex-linked inheritance. Whatever I explained to you, it was first discovered in only Drosophila melanogaster by scientist Thomas Morgan. I told you about T.H.Morgan. He is named or popularly as the modern geneticist or the experimental father of experimental genetics. Right. Again and again using the same word. Remember this. Right. Coming to the first thing, what he did was he was growing Drosophila in a group. Then all the Drosophila insects normally will have red colored eye, not color blindness. Don't mix with the color blindness and eye color in Drosophila. These Drosophila have the eye color which is red in color. The red eyed are dominant. But when he was growing the insects in a group, he found suddenly few male offsprings which are having white eye originated. He found that some were male insects, and when he checked their sex, they were all males. Then he gave the word that this character of eye color in Drosophila must be linked with the sex chromosome. It is a body character, right? Eye color and hemophilia, all they are the vegetative or somatic characters that might have linked with the X chromosome was his first explanation. Later he worked on that and he was the first person to explain sex-linked inheritance. Right? Therefore, whenever I talked about sex-linked inheritance with hemophilia, but first example should be in Drosophila. I just gave you hemophilia example because it will become more familiar to you, and because I am explaining in humans you will understand easily. But otherwise, first work was done in Drosophila melanogaster only by T.H.Morgan. So what is the character I told you? The red eye color P1 he took as a red-eyed female, which is because of wild variety. Earlier I told you wild means they are the original ones, crossed with the white-eyed males. Okay. What is the genotype of this? Genotype will be X capital R, X capital R if you consider, because it is a dominant character, wild character. So it should be capital R crossed with the white character X small r, X small r. Write gametes. Let me check how you write the gametes. So what are the gametes here? This female will produce only one gamete, this female insect. This male will produce not X. Male means X small r and Y, right? Here it will produce X small r one gamete, Y another gamete. Fertilization, X capital R, X small r. This is going to become, this is F1 generation. This is a red-eyed female. But what is it? It is a carrier. If this is the cross, X capital R and Y, this will be a red-eyed male normal, right? So P1 generation there was a white eye color. In F1 generation there is nothing called white. White color is not at all there. But white-eyed gene is located in the carrier female which is looking red-eyed. Now, when this female, red-eyed female of F1 get crossed or it is crossed with red-eyed male, what happens? Genotype, write the genotype X capital R, X small r, that one I am taking. F1, so remember F1 I am taking, crossed with the red-eyed X capital R and Y. What is the gametic generation? This will produce X capital R one gamete, X small r one gamete, crossed with X capital R one gamete, I am sorry, X capital R one gamete and Y is the another gamete. Now in F2 generation, you will get the checkerboard like this. Okay, write the big checkerboard. I told you mine is the place constriction I am writing. Constraints are there for me, I can't write big one here. Right? This is the male gamete, this is the female gamete. X capital R and Y is the male gamete. You have to take this. This is X capital R, females are this way, X small r. Fertilize X capital R, going to form the red-eyed female. X capital R and Y, it is the red-eyed male. X capital R, X small r, this is the red-eyed female, but she is a carrier because she is heterozygous. X small r and Y, it is the white-eyed male. Okay. Therefore, again F2 ratio what you will write? 3:1, among which males will have 1:1 ratio. This is very important. Observe this and try to write on your own without looking at the checkerboard, whatever I written. This also is showing crisscross inheritance. It was first identified in this organism only, Drosophila melanogaster. Did you follow? Take it down properly and write the analysis. Students, once we have finished with all the three examples of X-linked inheritance. See, I am using the word X-linked now because I told you sex-linked inheritance, major thing we learn those genes that are associated or linked with the X chromosome only. But now I will give you some important thing, very interesting thing, that is called Y-linked inheritance. What is Y-linked inheritance? This is the condition, as I told you, Y chromosomes will not contain the genes exactly that are located on X. Its same alleles are not there on the Y, but there is a small portion which is matching, but it will have individually some genes. There are certain body character identifying genes that are located only on Y chromosomes. Those genes that are linked with the Y chromosomes are called Y-linked genes. The genes that are linked with the Y chromosomes are called Y-linked genes. These inherit singly, one only. There is nothing another one present. They are anyway homozygous, heterozygous. Not only that, they are called hemizygous for these genes. And these genes that are linked with the Y chromosome inherit singly. This is called as a Hollandric gene. Remember, these are called the Hollandric genes. What is a Hollandric gene? The gene that is single and it inherits singly, normally attached or linked with the Y chromosome is called Hollandric gene. Because of this only we call Y chromosome the Hollandric chromosome also. Now, which character is linked with the Y chromosome in humans? There is no disease here. There is one character. We call that name. You remember the name of that character is hypertrichosis. Whole thing is single word, hypertrichosis. What is this then? Right? This is not a disease I told you. What is hypertrichosis? This is your common observation, you just do it. Most of the men, means not boys, men means around 30 to 40, that age, they develop certain hairs in their external ear pinna, means the hair come out from the ear pinna like this. This character is called hypertrichosis, right? This is not, the child will not bear the hair. Hair, it is a late secondary sexual character. It happens normally after 25 plus only. The development of hairs in the external ear pinna, this word you have to write, this sentence you should write, is called hypertrichosis. What is it? It is the type of Y-linked character in which men develop hairs in their external ear pinna. Internally they will come like this, right? That is called the hypertrichosis. It is a character in men. And now, it is linked with Y chromosome I told you. Then how it is inherited? If the father has hypertrichosis, he has hairs. If the husband and wife produce a child who is a male child, if they get a son, that son will get those hairs during in the external ear pinna when it will attain around 25 years or 30 years. Means hypertrichosis is directly transferred from the father to the son, right? Son to the grandson, right? Suppose the couple who are, the man is having hypertrichosis, female, they get married and they produce only girls, only they have daughters, the character will stop, right? There is no expression of this character. No females can get hairs in the external ear pinna. They may develop the hairs on the face. Facial hairs may develop sometimes in the females. because of the hormonal variation. Estrogen, progesterone, FSH, LH and all, some variation happens. Sometimes females develop hairs on the small moustache or beard like things they develop. This is just due to hormonal variations. But external ear pinna, they will never get hairs. Remember this. This is only restricted to men. Right? If the father has, son will get it. It goes in a straight line. No crisscross inheritance. Remember, hypertrichosis is not transferred through crisscross or the skip generation pattern. No. It will be directly transferred from parents to the offsprings and so on. That to male father to the son, son to the grandson and so on. Right? And one more thing some students will ask, what is the male other characters following? Y-linked inheritance, only one character we have, that is hypertrichosis. What more is called as the male pattern baldness. This is for the males it is. Pattern, we call it the male pattern baldness. Baldness means men after the age of around 45 plus getting bald is a dominant character. But that is not linked with the Y chromosome. Remember, it is because of the androgen, testosterone hormone. This is normal. The dominant character. 75% will get that male pattern baldness after 45 plus. Right? That is not connected to this. Y linked in it. But this also is a male character only. Pattern baldness is not seen in females. Females will not go bald. Very rarely they may get bald only if there is an infection in the scalp. Otherwise, normally females will not get baldness. They may lose hair, but they will not become bald. Right? Females can be happy now. Right? But males can become bald after 45. It is their natural character. It's a dominant character. They should see their parents, father, grandfather, if they have that baldness pattern, they are definitely going to get it. Right? Therefore, this genetics is like a fun you can study. Right? You have seen so many things I explained you. That I told you colorblindness. Colorblindness you are hardly, have you done that test? I showed you that this will have all the colors. Sometimes blue, green and the other colors also. This you have you watched any time in your, whenever you are getting, so many I have seen our students will get license. I don't know whether they get license or not, they will be driving vehicles. Do you check if you are going to get the license in soon, later after your age restriction, overcome the age restriction, then check this whether they have this kind of test with them in RTO office. If not there, you better insist there. I have seen some of my students who have gone as RTO officers, they have induced this because they came from science background. But mostly, and I have seen once this was test was there, I asked what is this, because they said colored Xerox is taking a lot of money, so we are converted to black and white print Xerox they have taken. How to identify color blindness? See, this is what we call science application. If you don't apply science properly, it is meaningless, right? Therefore, if such things you are not putting your brain, you are not putting science in that, that is a waste of getting literate also. As I told you, they did black and white Xerox and kept the paper. Do you have color blindness? Check this. What to check in that? Right? So it is simply meaningless. Therefore, use the science properly. Don't misuse science. Don't consider that science is something to joke. Right? This is basic requirement. Otherwise, it is life and death problem. Remember this. That is color blindness, which comes under sex-linked inheritance. So, I hope you are getting this subject perfectly well, and this almost completes Mendelian classical genetics. Next class, we are going to start with something molecular biology, which is more technical compared to this. Okay? Do well, study well, apply science wherever it is coming in your life, and see how scientific patterns happen in your life also, and educate people. Not only scoring, educate people about science. I told you, carrier females may be there. If somebody is not clotting blood, all these things you have to keep watching. Have a good vision. Then you can become good, healthy citizens. Okay, that is your basic requirement to learn science and spread size. Okay, see you with this.